Hi, my name is Aida and I,
And I'm Mirzakhanian.
and we will be presenting the RNA World. The RNA World hypothesis was coined by Walter Gilbert. He came up with the idea that evolution was based on RNA. Molecules such as ribose, phosphate, purines, and pyrimidions have been found to appear before the advent of life on Earth. RNA is abundant in all living cells. It can replicate easily in natural environments due to base pairing. DNA most likely evolved from RNA, and RNA has led to genetic code which produce proteins and eventually the formation of cells. RNA can also act as an enzyme to catalyze reactions necessary for life. Satisfies the requirement for natural selection. RNA can replicate. Differences in replication is reflected in the new strand. Process of RNA replication involves errors which lead to mutation, and differences in replication outputs lead to differences in frequency in the population. What is RNA? RNA is a molecule that acts as a messenger carrying instructions from DNA for controlling the synthesis of proteins. This consists of a long chain of nucleotides. Nucleotides are a compound of molecules that form the building blocks of DNA or RNA. This is composed of phosphate groups, the bases adenine, cytosine, guanine, and thymine, and a pentose sugar in RNA, the thymine base being replaced in uracil. Base pairing, a base of complementary bases in a double-stranded nucleic acid molecule consisting of purine and one strand linked by hydrogen bonds of pyrimidine in the other. Cytosine always pairs in guanine and adenine with thymine in DNA and uracil in RNA. In the picture on the right, you can see the difference between DNA and RNA. There are a few important experiments that demonstrate the plausibility of this hypothesis. In 1965, Spiegelman and his colleagues performed an experiment to uncover the origins of natural selection. In this experiment, they added nucleotide bases, which are adenine, guanine, cytosine, and uracil, and a replicase enzyme into a test tube and incubated the mixture. They discovered that natural selection acted on the RNA copies, which led to errors in the process of replication, and this led to mutations in which natural selection acted on. This led to changes in characteristics over time.
In 1975, Manfred Sumper and his colleagues further examined natural selection in the early stages of RNA-based life. In this experiment, scientists added acridine orange, a molecule that prevents replication. They discovered that RNA adapted to this change and was able to replicate. In fact, the new RNA strands replicated more efficiently in the presence of acridine orange.
In 2002, Natasha Paul and Gerald Royce demonstrated that RNA can act as an enzyme and catalyze reaction required for its own assembly. In 2009, Lincoln and Joyce designed an experiment to further test this hypothesis. In this experiment, RNA was provided with different substrates, which led to variants and multiple mutations. This experiment demonstrates how RNA might have been the first things for natural selection to act on and evolve. How did we go from an RNA world to DNA? DNA is chemically more stable than RNA, primarily because the deoxyribose sugar that composes DNA is less reactive than RNA's ribose sugar. Most importantly, DNA allowed for specialization within cells. And this concludes our presentation of the RNA world. Thank you.